Good day to viewers. Today we are going to discuss about the continuation of vectors, the plasmids. Plasmids or bacterial vectors. Let us get into details of the plasmids today. Plasmids are extrachromosomal DNA, usually circular in nature. These DNA encode auxiliary functions for the growth of the bacterium, which are essential for specific environments, virulence, antibiotic resistance, use of unusual nutrients, production of bacteriocins, etc. These plasmids must be a replicon. That means they should have a self-replicating genetic unit. DNA replication and partition are the key responsibility or key genes which are responsible for sending the plasmids to the next generation, which make sure each progeny cells receives a plasmid so that the genes are continued in the next generation. High copy number plasmids are usually small. Low copy numbers plasmids can be large. Partitioning is strictly controlled for low copy number, but it is loose for high copy number since the copy number is high. Plasmids are incompatible when they cannot be stably maintained in the same cell because they interfere with each other's replication. So when you have more than one type of plasmid in a cell, the plasmid compatibility and incompatibility has to be checked with the organisms. So this is an old school method for purifying your plasmid DNA, which is nothing but your cesium chloride gradient in the presence of ethidium bromide, which is going to illuminate the DNA in the presence of a UV light. So you have three different forms of a plasmid, which is detailedly given in this picture. And upon separation, the forms of plasmids are separated and the DNA is illuminated in the presence of your chemical substance ethidium bromide in the presence of ultraviolet light. These are some of the traits of the naturally occurring plasmids which have the plasmid col-E1, which is present in the bacterium Escherichia coli, as the gene for bacteriocin, which is going to help the bacterium to kill the other strains of E. coli. Tol gene, which helps the plasmid to degrade, helps the bacterium to degrade toluene and benzoic acid. It is isolated from Pseudomonas butida. And Ti plasmid, which is called as tumor initiation in plants. So it is present in the bacterium Agrobacterium tumefaciens. PJP4, which codes the gene for 2,4-D, that is dichlorophenoxyacetic acid degradation, and it is isolated from Alcaligens eutrophus. And PSIN, which is responsible for nodulation on roots of legume plants. It is isolated from the bacterium Rhizobium meliti. And SCP1, which is a gene which codes for antibiotic methylomycin biosynthesis. It is isolated from Streptomyces coelicola. RK2 is a gene responsible for resistance to ampicillin, tetracycline and kanamycin. So it is isolated from Klebsiella aerogenisa. So coming to the replication of the plasmid DNA, plasmid replication requires host DNA replication machinery. Wild plasmid carry genes needed for the transfer and copy number control. All self-replicating plasmids have an origin of replication. Some plasmids also carry ORIT, which is also called as origin of transfer. So they carry plasmids which carry function needed to be mobilized or from one bacterium to other bacterium. Plasmid segregation is maintained by a paralocus, a partition locus that ensures each daughter cell gets on plasmid, and not all plasmids have such sequence. There are 5 main incompatibility groups in your plasmid replication. Not all plasmids can live with each other in the bacterium. So plasmid incompatibility is another key parameter which has to be decided for bacterial cloning or gene transfer. Agents that disturb DNA replication or destabilize or cure plasmids from cells are also present. So as I've already said you, incompatibility groups, not all plasmids can live together. Plasmids that are able to coexist in the same cell do not interfere with each other's replication. A single cell can have many inclusive group of plasmids as it can tolerate and replicate. The partition locus, a region on the broad host range plasmid that binds to a structure on the inner membrane of the cell to ensure proper segregation. Plasmids labeled with fluorescent protein move to each daughter cell during the process of cell division, thereby ensuring that each daughter cell gets a copy of the given plasmid. So thank you for your patient listening. We have seen about plasmids and their characteristics. Let's get into the artificial plasmids with examples in the next lecture. Thank you.